Hi class, this is your instructor, Skylar Huff. We're now in Chapter 13, The Chromosomal Basis of Inheritance and Human Genetics. Mendel's experiments opened the door to understanding inheritance, but many questions remained. In the early part of the 20th century, we did not know the nature of the factors whose behavior Mendel had described. The next step, which involved many researchers in the early part of that century, was uniting information about behavior of chromosomes seen in the picture and the inheritance of traits. This led to a deeper understanding of Mendelian genetics and opened the way for such techniques as genetic mapping, which are still important today. With this expanded view of Mendelian genetics, we will explore human genetics in some detail. We will see how humans are not that different from our model organisms and how human genetics has become one of the most exciting fields of biology. So in this chapter, we will not go through each section. We'll go through the first section and the last two sections. Let us begin. A role for chromosomes in heredity was first suggested in 1900 by the German geneticist Karl Correns in one of his papers announcing the rediscovery of Mendel's work. Soon afterwards, observations that similar chromosomes paired with one another during meiosis led directly to chromosomal theory of inheritance, first formulated by the American Walter Sutton in 1902. In 1910, Thomas Hunt Morgan, studying the fruit fly Drosophila melanogaster, discovered a mutant male fly with white eyes instead of red. You can see that here in Figure 13.1. Morgan immediately set out to determine whether this new trait would show Mendelian inheritance. He crossed the mutant male to a normal red-eyed female to produce an F1 with all red eyes, leading Morgan to conclude that red eye color was dominant over white. Morgan then crossed red eye flies from the F1 generation with each other. Of the 4,252 F2 progeny, Morgan examined 782, which is about 18%, had white eyes. Although the ratio of red eyes to white eyes in the F2 progeny was greater than the 3-to-1 ratio, the results nevertheless provided clear evidence that eye color segregates. However, something about the outcome was strange and totally unpredicted by Mendel's theory. All of the white-eyed F2 flies were males. So please see Figure 13.2, which is here. White eyes in the male flies by way of the F2 generation. One possible explanation was simply that for some unknown reason, white-eyed female flies are not viable. To test this idea, Morgan test-crossed the female F1 progeny with the original white-eyed male. He observed white-eyed and red-eyed flies of both sexes in the expected 1-to-1 ratio, also shown in Figure 13.2, which shows that white-eyed female flies are viable. This led Morgan in a more interesting direction as he looked for an explanation in the actual chromosomes in males and females. In Drosophila, the sex of an individual is determined by the number of copies it has of a particular chromosome, the X chromosome. Observations of Drosophila chromosomes revealed that female flies have 2 X chromosomes, but male flies have only 1 X chromosome. In males, the single X chromosome pairs in meiosis with a dissimilar partner called the Y chromosome. These 2 chromosomes are termed sex chromosomes because of their association with sex. The solution to Morgan's puzzle is that the gene causing the white-eyed trait in Drosophila resides only on the X chromosome. It is absent from the Y chromosome. A trait determined by a gene on the X chromosome is said to be sex-linked or X-linked because it will segregate with the sex chromosomes. Because white is recessive, males carrying an X chromosome with this allele will always appear white-eyed. Morgan's result was a natural consequence of the Mendelian segregation of chromosomes. And what he had actually shown was that the segregation of a visible trait correlated with the segregation of a chromosome. One of Morgan's students, Calvin Bridges, later showed that aberrant segregation of X chromosomes also correlated with the aberrant segregation of the white gene. This is considered the final proof of genes being on chromosomes, as both normal and abnormal segregation of a chromosome pair could be perfectly correlated with normal and abnormal segregation of a trait. The structure and number of sex chromosomes vary in different species. This is shown to you all in Table 13.1. Please look at it. In the fruit fly, Drosophila, females are XX and males XY, which is also the case for humans and other mammals. However, in birds, the male has 2 Z chromosomes and the female has a Z and a W chromosome. Some insects, such as grasshoppers, have no Y chromosome. Females are XX and males are characterized as XO. O indicates the absence of a chromosome. In Chapter 10, you learned that humans have 46 chromosomes, 23 pairs. One pair consists of the sex chromosomes, which are dissimilar, and the other 22 pairs, which are similar, are called autosomes. The Y chromosome in males is highly condensed. Because few genes on the Y chromosome are expressed, recessive alleles on a male's single X chromosome have no active counterpart on the Y chromosome and form a new kind of genotype, hemizygous. The default setting in human embryonic development leads to female development. Some of the active genes on the Y chromosome are responsible for the masculinization of genitalia and secondary sex organs, producing features associated with maleness in humans. Consequently, any individual with at least one Y chromosome is normally a male. In vertebrates, this form of sex determination is seen in mammals and birds, is called chromosomal sex determination. Although males have only one X chromosome and females have 2, female cells do not produce twice the amount of proteins encoded by genes on the X chromosome. Instead, one of the X chromosomes in females is inactivated early in embryonic development, shortly after the embryo's sex is determined. This inactivation is an example of dosage compensation, which ensures an equal level of expression from the sex chromosomes despite a differing number of sex chromosomes in males and females. In Drosophila, by contrast, dosage compensation is achieved by increasing the level of expression on the male X chromosome. Which X chromosome is inactivated in human females varies randomly from cell to cell. If a female is heterozygous for a sex-linked trait, some of the cells will express one allele and some the other. The inactivated X chromosome is highly condensed, making it visible as an intensely staining bar body. And if you look in your text, you can see that shown. It is not shown here. And the bar body is attached to the nuclear membrane. The X chromosome inactivation mechanism of dosage compensation is not unique to humans, but is true of all mammals. Females that are heterozygous for X chromosome alleles are genetic mosaics. Their individual cells may express different alleles depending on which chromosome is activated. One example is the calico cat, a female that has a patchy distribution of dark fur, orange fur, and white fur, shown here. The dark fur and orange fur are due to heterozygosity for one gene on the X chromosome that determines pigment color. One allele results in dark fur, and another allele results in orange fur. Which of these colors is observed in a particular patch is due to inactivation of one X chromosome. If the chromosome containing the orange allele is inactivated, then the fur will be dark, and vice versa. The patchy distribution of color and the presence of white fur is due to a second gene that is epistatic to the fur color gene. That is, the presence of this second gene produces a patchy distribution of pigment, with some areas totally lacking pigment. In the areas that lack pigment, the effect of either fur color allele is masked. Thus, in this one animal, we can see an excellent example of both epistasis and X inactivation. Now we'll move on over to human genetics. With the discovery of Mendel at the beginning of the 20th century, there was a rapid growth in knowledge of the basic mechanisms of heredity. This was driven by researchers concentrating on the number of model systems that were considered to be genetically tractable. Short generation times, ease of performing controlled crosses, and the ability to induce and maintain collections of mutants. This included fruit flies from Drosophila, yeasts Saccharomyces cerevisiae, the mouse Musculus, and then worms from the epithet elegans, so it says C. elegans, and then Zea mays, which is corn, and a small flowering plant researched so much Arabidopsis thaliana. The information from these model systems was assumed to be relevant to humans, but there was not a lot of direct information about human genetics. Since the discovery of Mendel's work, human geneticists have looked for traits that follow simple Mendelian inheritance. A number of traits were recognized fairly quickly, which included both dominant and recessive modes of inheritance. Many human traits are also affected by multiple genes, and human genes can affect more than one phenotype. In other words, human genetics appears to show the same features discovered in model systems. Simple recessive traits range from those that affect appearance, like albinism, to those that can result in serious clinical syndromes like cystic fibrosis and Tay-Sachs disease. It should also be noted that genes with variants that result in albinism in humans are evolutionarily related to albinistic genes in other mammals as well. Simple dominant traits behave in humans similarly to those traits in our model organisms and can have effects that range from mild to severe. Even the alterations of dominance we discussed in Chapter 12 are seen in humans. For example, while there are many genes that affect hair morphology, there is also a gene that affects curly versus straight hair and displays incomplete dominance. In this case, a homozygous dominant individual has curly hair, while a heterozygous individual has wavy hair, and a homozygous recessive individual has straight hair. Since researchers cannot perform controlled crosses in humans, as in our model systems, we instead look to crosses that have already been performed. In other words, family histories. The organized methodology we use is a pedigree, a consistent graphical representation of matings and offspring over multiple generations for a particular trait. In a pedigree, males are shown as squares and females as circles. Those showing a trait usually referred to as affected In the case of disease states are shown with shaded squares or circles, and those unaffected are shown as unshaded. The information in the pedigree may allow geneticists to deduce a model for the mode of inheritance of a trait. In analyzing pedigrees, it is important to remember that disease-causing alleles are usually rare in the general population. If it is necessary to assume too many unrelated affected individuals, your model is probably wrong. An example of a dominant pedigree for juvenile onset glaucoma is shown in figure 1310. And that is being shown here now. This is figure 1310. One feature that is immediately obvious is that the trait tends to appear in every generation, and affected offspring always have at least one affected parent. It is also possible in a dominant pedigree for two affected parents to have unaffected offspring, which would not be possible with a recessive trait. An example of a recessive pedigree for albinism is shown in figure 1311. That is now shown. In cases where genotypes of heterozygotes who do not display the trait are known or can be inferred, they are shown as half-shaded. The hallmark of a recessive pedigree is that affective individuals can result from two unaffected individuals, which cannot occur in a dominant trait. For recessive traits, one affected parent usually does not result in affected offspring, and the trait will be equally distributed among males and females. From ancient times, people have noted conditions that seem to affect males to a greater degree than females. This is easily explained if the genes responsible are located on the X chromosome. Red-green color blindness is a well-known condition that is more common in males because the gene affected is carried on the X chromosome. Another example is hemophilia, a disease that affects a single protein and a cascade of proteins involving the formation of blood clots. Thus, in an untreated hemophiliac, even minor cuts will not stop bleeding. This form of hemophilia is caused by an X-linked recessive allele. Women who are heterozygous for the allele are asymptomatic carriers, and men who receive an X chromosome with the recessive allele exhibit the disease. The inheritance of sex-linked traits shows some interesting patterns. Sons always get an X chromosome from their mother, so any sex-linked disorders come from their grandfather through their mother, not their father. Heterozygous mothers are referred to as carriers. This leads to these traits often skipping a generation. Females can have X-linked disorders, but only if their father was affected and their mother was a carrier. It is instructive to examine a single disorder in detail to see how a single amino acid change in a single protein can result in a clinical syndrome and can have evolutionary implications. The first human disease shown to be caused by a mutant protein was sickle cell anemia, which causes intermittent illness and reduced lifespan. It is caused by a defect in the oxygen-carrying molecule hemoglobin. Sickle cell hemoglobin molecules tend to aggregate inside red blood cells, forming rod-like structures that distort the shape of cells and reduce their ability to transport oxygen. These red blood cells have a characteristic shape that led to the name sickle cell, as shown here in figure 1312. Individuals heterozygous for the sickle cell allele are indistinguishable from homozygous dominant individuals in a normal oxygen environment. Although their red cells do exhibit reduced ability to carry oxygen. The sickle cell allele is not the only structural variant of hemoglobin. In fact, over 700 have been cataloged. It is estimated that 7% of the human population worldwide are carriers for different inherited hemoglobin disorders. This includes thalassemia, which show altered expression of globin genes and are not structural changes. The sickle cell allele is particularly prevalent in people of African descent. In some regions of Africa, up to 45% of the population is heterozygous for the trait, and 6% are homozygous. This proportion of heterozygotes is higher than expected based on chance alone. It turns out that heterozygosity confers a greater resistance to the blood-borne parasite that causes malaria. In regions of Central Africa, where malaria is endemic, the sickle cell allele also occurs at a high frequency. Table 13.2 provides additional examples of selected genetic disorders. I recommend you see Table 13.2 to prepare well for your test. This is obviously a very short list and raises the question of how many Mendelian phenotypes exist in humans. One approach to answer this is to use the mouse, a fellow mammal, as a model system. So in Table 13.2, it gives you the disorder. It gives you the symptom, the defect, whether it's dominant or recessive, and even whether it's X-linked or not, and the frequency among human births, not to mention particular groups of people who are more affected than others with those disorders that are genetic. So back to what I mentioned about the mouse. Loss of function mutations were constructed in 900 mouse genes and analyzed for a phenotype. At least 30% of the mutants exhibited embryonic lethality, and the majority had at least one phenotype. This implies that the majority of human genes could potentially produce a phenotype when mutated. Information about human genetic diseases and genetic variants can be found in the many databases dedicated to making these data accessible. The oldest is Online Mendelian Inheritance in Man, or OMIM. This effort begun in 1966 with the publication of Mendelian Inheritance in Man by Victor McCusick. After multiple print editions, it was moved online and made publicly available in 1987. This was followed by moving onto the then young World Wide Web in December of 1995. Summary statistics from 2020 indicate that there are 6,771 phenotypes with known molecular bases. From the perspective of genes, there are 4,355 genes with mutations that cause phenotypes. Among these genes, many produce multiple phenotypes. 794 with 2 phenotypes, 293 with 3, and 238 with 4 or more phenotypes. So here is that table mentioned, Table 13.2, with the disorder, symptoms, the defect, dominant, recessive, or X-linked, the frequency among human births, and those affected groups of people as mentioned just moments ago. So far, we've been considering only single gene disorders. There are also clinical syndromes due to changes in chromosome number. The failure of homologs or sister chromatids to separate properly during meiosis is called non-disjunction. This failure leads to the gain or loss of a chromosome, a condition known as aneuploidy. The frequency of aneuploidy has been estimated at 7 to 10% of clinically recognized conceptions. Individuals who have lost one copy of an autosome are said to be monosomic, and generally do not survive embryonic development. In all but a few cases, individuals who have gained an extra autosome called trisomics also do not survive. Data from clinically recognized spontaneous abortions indicate levels of aneuploidy as high as 35%. Embryos trisomic for 5 of the smallest human autosomes, numbers 13, 15, 18, 21, and 22, can develop and survive to live birth. Trisomy 13, 15, or 18 causes severe developmental defects in infants. With such a genetic makeup, die within a few months. In contrast, individuals trisomic for chromosome 21 or more rarely chromosome 22 usually survive to adulthood. And that says rarely. In these individuals, the maturation of the skeletal system is delayed, so they generally are short and have poor muscle tone. Their mental development is also affected, and children with trisomy 21 show some degree of intellectual disability. The developmental defect produced by trisomy 21 was first described in 1866 by J. Langdon Down. For this reason, it is called Down syndrome. About 1 in every 750 children exhibits Down syndrome, and the frequency is comparable in all racial groups. Similar conditions also occur in chimpanzees and other related primates. Only a portion of chromosome 21 needs to be present in 3 copies to produce the clinical syndrome. In 97% of the cases examined, all of chromosome 21 is present in 3 copies. In the other 3%, a portion of chromosome 21 containing the critical segment has been translocated to another chromosome. It exists along with the normal 2 copies of chromosome 21. This latter condition is known as translocation Down syndrome. There is an association between maternal age and the risk of Down syndrome. For a maternal age of 20 years, the risk of Down syndrome is 0.07%. With maternal age of 30, the risk rises to 0.1%. And with a maternal age of 40, the risk is 1%. Chromosomal abnormalities arise more frequently on the female side, where gametes are set aside early in development, and the rest in meiosis I with homologs paired. This is also thought to be the molecular basis of the increase in aneuploidy with maternal age. So what you're seeing here is figure 1313, and this is a male karyotype. And if you look at chromosome number 21, or at least the 21st position, or autosomes 21, you will see it is trisomy 21, the trisomic condition. So this is showing the risk for Down syndrome and the percentage of affected individuals among live births. So there is a direct correlation between advanced maternal age and the incidence of Down syndrome, as just mentioned. Aneuploidy for the sex chromosomes has less severe consequences than autosomal aneuploidy. The specific sequences depend on genotype, but individuals may have normal features, often reach maturity, and in some cases may be fertile. Non-disjunction of sex chromosomes can occur in either male or female meiosis. In female meiosis, this will produce oocytes with either 2 X chromosomes or no X chromosome. In male meiosis, the outcome depends on whether the non-disjunction occurs during meiosis 1 or 2 and can produce XY sperm, YY sperm, and sperm with no sex chromosomes. Various combinations of these gametes and normal gametes leads to zygotes with sex chromosome aneuploidies. So you have XXX, XXY, XO or OY, and XYY. The most extreme case is the complete lack of an X chromosome, OY, which results in embryonic lethality. At the other end of the spectrum, XXX individuals develop into females with 1 functional X chromosome and 2 bar bodies. They may be taller in stature, but are otherwise indistinguishable from XX females. This condition, called XXX, occurs in about 1 per 1,000 females. Similarly, XYY zygotes develop into fertile males of normal appearance. The frequency of XYY genotype, called Jacob syndrome, is about 1 per 1,000 males. The effects are more serious for XXY and XO zygotes. An XYY individual develops as a male with many female body characteristics and in some, but not all cases, diminished mental capacity. This condition, called Klinefelter syndrome, occurs in about 1 per 1,000 male births. XO zygotes will develop into a sterile female of short stature with a webbed neck and sex organs that never fully mature during puberty. The mental abilities of an XO individual are in the low to normal range. This condition, called Turner syndrome, occurs roughly once in every 5,000 female births. This shows non-disjunction and sex chromosomes. Although most genetic disorders cannot be cured, progress toward successful therapies is being made in many cases. In the absence of therapy, one approach to cope with genetic diseases is prevention. Genetic counseling can help to identify parents carrying disease alleles, and prenatal testing can assess the genetic state of early embryos. Pedigree analysis is one way of assessing risks, often employed as an aid in genetic counseling. For example, if a counseling client's family history reveals that a relative has been afflicted with a recessive genetic disorder, such as cystic fibrosis, it is possible that the client is a heterozygous carrier of the recessive allele for that disorder. If a couple is expecting a child and the pedigree analysis indicates that both of them have a significant chance of being carriers of a deleterious recessive allele, the pregnancy is said to be high risk. In such cases, a significant probability exists that their child will exhibit the clinical disorder. Another class of high-risk pregnancy is that in which the mothers are older than 35. As discussed earlier in this section, the frequency of Down syndrome increases dramatically in the pregnancies of older women, and that was shown here. When a pregnancy is considered high risk, many women elect to undergo amniocentesis, a procedure that permits the prenatal diagnosis of many genetic disorders. In the 4th month of pregnancy, a sterile hypodermic needle is inserted into the expanded uterus of the mother, removing a small sample of amniotic fluid that bathes the fetus. This fluid contains fetal cells that can be grown in culture for analysis. During amniocentesis, the position of the needle and that of the fetus are usually observed by means of ultrasound, which is not harmful to the mother or fetus and aids in the withdrawal of amniotic fluid without damaging the fetus. In addition, ultrasound can be used to examine the fetus for signs of major abnormalities. However, about 1 in 200 amniocentesis procedures result in fetal death and miscarriage. chorionic villus sampling. It's a less invasive procedure for prenatal diagnosis. This method involves removing cells from the chorion, a membranous part of the placenta that nourishes the fetus. This procedure can be used earlier in pregnancy, by the 8th week, and yields results much more rapidly than does the amniocentesis. Risks from chorionic villus sampling are comparable to those for amniocentesis. To test for specific genetic disorders, genetic counselors look for 3 characteristics in the cultures of cells obtained from amniocentesis or chorionic villus sampling. First, analysis of the karyotype can reveal aneuploidy and gross chromosomal alterations. Second, in many cases, we can test for the proper functioning of the affected enzyme. For example, the absence of the enzyme responsible for the breakdown of gangliosides is diagnostic for Tay-Sachs disease. Third, DNA can be directly analyzed for lesions known as genes. In that last case, the direct analysis of DNA, the number of genes that are known to produce a Mendelian phenotype has exploded in recent years, while diagnostics lag behind gene discovery. It is still possible to test for a large number of disease variants. Both amniocentesis and chorionic villus sampling are considered to be invasive procedures. The discovery that a number of biomarkers found in maternal blood plasma could be correlated with Down syndrome led to non-invasive screening methods for this particular trisomy. More recently, the fetal DNA in maternal blood plasma, which is cell-free DNA or cfDNA, has led to new methods of non-invasive prenatal screening. At present, these tests can be used to screen for trisomy of chromosomes 13, 18, and 21, and used for sex determination. Considerable effort is being put into expanding the scope of this type of screening, and it is difficult to predict how fast or how far it will go. It should be remembered that this form of screening and is not at present diagnostic. I repeat, this is a form of screening and is not at present diagnostic. A positive result will indicate the need for more invasive techniques for diagnosis. And presently, only diagnosis of paternally inherited conditions due to DNA not present in the maternal genome is possible. In model systems, geneticists analyze biological systems by first inducing mutants, selecting for relevant phenotypes, then finding the genes responsible for these phenotypes. This involves placing genes on genetic maps to determine their relative positions in the genome, then using molecular cloning to isolate the gene or genes of interest. In some systems, it is possible to start with a gene of unknown function, disrupt the gene, and replace the normal copy in the organism to assess its function based on phenotype. This is the opposite logic of traditional analysis. It has been called reverse genetics. To put in simple terms, forward genetics goes from phenotype to genotype, and reverse genetics goes from genotype to phenotype. In studying human genetics, we obviously cannot use either of these approaches directly, but we can apply the same logical approaches to and use the data we have, pedigree data from families, and the enormous variation found in human populations. When genetic maps were first constructed in experimental organisms, the majority of genetic markers caused a visible phenotype, such as changes in eye color in flies. In humans, the equivalent genes cause disease states. These do not make good genetic markers because of their low frequency in the population and because few were known in the early days of human genetics. In the early 1980s, the number of markers for the human genome numbered in the hundreds. This situation changed with the identification of differences in DNA between individuals that do not alter phenotype. One widely used kind of marker is short tandem repeat, or STR. These are short repeats of 2 to 4 bases that differ in the repeat number between individuals. These have been used in genetic mapping and also form the basis for databases used in the forensic analysis of DNA. Molecular markers have evolved with technology and allow the production of human genetic map that would have been unthinkable 30 years ago. So short tandem repeats. And one more thing I'll show is figure 3.17. So this is only a partial map for the human X chromosome. A more detailed map will require a much larger figure. So the black bands represent the staining patterns that can be seen under a microscope, and the constriction represents the centromere. The analysis of the sequence of X chromosome indicates 1,805 genes on the X chromosome, and 821 of these have mutant alleles that have been identified as affecting disease states. Of these, 59 diseases are shown that have been mapped to specific segments of the X chromosome, indicated by brackets. Using data from sequencing the human genome allows the identification and mapping of single base differences between individuals. Any differences between individuals in populations are termed polymorphisms. Polymorphisms affecting a single base of a gene locus are called single nucleotide polymorphisms, or SNPs. There are now more than 600 million single nucleotide polymorphisms identified in the human genome. The vast majority of these are at very low frequency in populations, with only about 10 million at a frequency of greater than 5%. These have been identified and mapped by an international consortium, with data being publicly available. With these common molecular markers in hand, a dense genetic map was constructed as part of the Human Genome Project. This genetic map provides a framework to locate genes of interest, that is, those that affect interesting phenotypes. Human genes can be mapped similar to genetic mapping in model organisms, but using data derived from families instead of controlled crosses. The principle is that the same genetic distance is still proportional to recombination frequency, but the analysis requires the use of complex statistics and summing data from many families. In addition to genetic maps, there are a variety of physical maps that can be constructed as well. The combination of genetic and physical mapping was an integral part of the Human Genome Project. Here we would consider the use of genetic mapping and association studies in human genetics. The power of genetic mapping is that it allows geneticists to find the genes responsible for specific phenotypes. Even before the Human Genome Project was completed, geneticists were finding genes that could be mutated to cause particular genetic diseases, including cystic fibrosis, the recessive allele, and Huntington's disease, the dominant allele. Linkage analysis can identify rare casual variants in a population that have a large effect on phenotype. This limits gene discovery using linkage analysis to genetic disorders caused by a single gene, so-called monogenic disorders. The logic is to follow the segregation of possible casual alleles with disease state in multiple families. The more data available, the smaller region of the genome this can identify. While the process is laborious and time-consuming, by the time of publication of the sequence of the human genome, around 1,000 single-gene inherited diseases had been characterized. Where linkage-based analysis is not effective is in determining the genes involved in complex traits affected by many genes. The advent of the data from the Human Genome Project led to the possibility of analyzing complex traits with no single variant with a large effect. Although there are an estimated to be some 7,000 inherited diseases caused by rare variants of single genes, most human phenotypes, including many common diseases, are affected by many genes. We saw in the last chapter that complex traits are usually so-called quantitative traits that are due to the action of many genes. To address these traits, a different methodology was needed. Although still a phenotype to genotype forward genetics approach. So thus the Genome-Wide Association Study, or GWAS, was born in 2005. In these studies, it is possible to start with a complex disease phenotype, for example, coronary artery disease, then look for single nucleotide polymorphisms associated with this phenotype in a population. As the location of each single nucleotide polymorphism is known, you can look for candidate genes in that region of the genome. Coronary artery disease is an interesting example, as few variants were identified with a relatively large effects. One variant found in both U.S. and European populations is the most common genetic risk factor and accounts for more than 10% of disease risk. But this and other variants with large effects do not account for the majority of heritability. There are literally thousands of variants with small effects that contribute to coronary artery disease and account for the majority of the heritability. This pattern has held true for most complex diseases analyzed by GWAS, the Genome-Wide Association Study. GWAS have also been used to look at complex traits that have high heritability, such as height and body weight. For height, over 700 common variants, a frequency over 5% in the population, have been shown to affect height, each with a relatively small effect. The smaller number of rare variants have also been identified, but detection requires analyzing very large populations. Most GWAS to date have used techniques that involved analyzing single nucleotide polymorphisms and are thus limited by the number of single nucleotide polymorphisms assayed. The more rare variants that are used, the more that are found to have small effects. At this point, while they have provided a mountain of data, over 3,000 studies, GWAS are still a young technology. Despite this, they are changing how we view human phenotypes. Complex human phenotypes appear to be affected by thousands of relatively common genetic variants, each with small individual effect, which are predominantly found in non-coding regions. These most likely affect gene expression, and thus a single variant can affect multiple phenotypes. Rare variants affect human phenotypes as well, but their identification is hampered by the size of populations required to produce sufficient statistical power. It is important to emphasize that finding a variant that is associated with phenotype does not mean the variant itself is causal. The variant may be close to a gene that affects a phenotype, but the variant itself does not, or the variant may affect the regulation of a gene with a direct effect. The greatest limitation of these studies is that they tell us nothing about the function of the variants identified. Both linkage analysis and GWAS use logical forward genetics, that is, going forth from a known phenotype to an unknown genotype. The only mammalian system that has powerful techniques of reverse genetics is mouse. The ability to knock out genes in the mouse is a powerful use of reverse genetics to discover the function of specific genes. While this is not possible in humans, we can look for potential loss of function alleles, such as ones that result in drastically shortened proteins, which are expected to lack function. This work is still in the early stages, but putative loss of function variants for those thousands of genes are being identified. These are mainly at low frequency, less than 0.1% in the population. So the number that are homozygous is extremely low, but these represent genes that can be considered non-essential, although of still unknown function. Despite the early state of these data, around 1,800 genes have been identified as having loss of function variants that can exist as homozygotes. The next step in this approach is to correlate homozygous loss of function variants with possible phenotypes. At least one group is moving towards a human knockout gene project. Another approach is to take advantage of the large databases containing phenotypic data linked to genotypic data. Some electronic health records are being designed to standardize phenotypic data, and when these include volunteers to provide DNA for analysis, this allows a form of reverse genetics in humans. This kind of population-based analysis, the Phenotype-Wide Association Studies, PHEWAS, uses known variants to assess their association with a wide spectrum of phenotypes in the electronic health records. The PheWAS can reveal the function of unknown variants and begin to unravel how many variants affect different phenotypes. This can also be used to identify variants that affect more than one phenotype, or pleiotropy. Early results indicate that many common variants affect multiple phenotypes, implying high levels of pleiotropy. The next step is to unify the approaches of GWAS and PheWAS to explore both sides of the genotype-phenotype relationship.